Today we will be going with the second line of defense. As we were done with the first line of defense, now what's the second line of defense? That means like if bacteria has crossed the first line of defense, so it will enter in the second line of defense. Okay, so as we were done with the first line of defense, here let me show you. In that we were done about the physical barrier, chemical weapons, and as we know, like these were non-specific, means they were not working on a particular or specific one target. That's the reason they were non-specific. So now let's discuss second law of defense in detail. So let's start with this. So second line of defense, we were having killer cells, like which is having types, which is monocytes, NK, which is known as natural killer cell, and neutrophils, cytokine, which were having protective proteins, fever, inflammation. This means if the first bacteria cross the first barrier, so it will enter in the second line of defense, in which we will get very mild fever, like fever, inflammation would be there. So as bacteria will cross first line, it will enter in the second line. So here we are having major components which will be discussed here. Major components, which are killer cells, fever, and inflammation. These three are the major components of second line of defense. So first, this killer cell will be separated or divided into monocytes and dendrite cells. So monocyte, which is like type of white blood cell, and when in white blood cell, it's the type of leukocytes. Okay, cover approximately 2 to 8% of white blood cell. Even this value may varies book to book, like it can be 2 to 5, but approximately it would be 2 to 8. So then there's ag- granulocytes. Granulocytes, these are like pigmented molecule. They're absent in nucleus. They are present in bone marrow. Originated from myeloid. Sorry, wait. Originated from myeloid stem cells. These monocytes are present in a bone marrow, and they are originated from myeloid stem cells, but they will be present after that. They will be present in blood for the maturation, like they will get matured there, maturation process, and then their name would be changed. That would be macrophagocytes. Now what's their work? Or now what is this macrophagocytes? Like this is divided into two. Macro means big, and phagocytes means eat. It's mean after that they're very freely to move anywhere. They can move from one part to another. Like they are having, we can say they are having characteristics to move now, or they got an ability to move now anywhere where they want, even in the vital organs. So now let's see. So now macro As we discussed, like they can go in any location or like freely to move. Like they are the largest cell of like all other leukocytes. Like these are the largest cells in all of the leukocytes. Then we are having monocytes and macrophagocytes, which like they form. So these macrophagocytes are like, as I told, these are freely to move anywhere. They are having horseshoe structure, like horseshoe shape nucleus, and their lifespan is like can be months and like years, how much body need, or like it would stay as long as that went. And the life of this macrophagocyte would be 100 pathogens at a time. So this is the another characteristic of macrophagocytes. Macrophagocytes will work as a protection. Wait, let me write. They will work as as a protection of our vital organs. So now let me tell you the difference of organs and vital organs. So like vital organs are those organs which are very essential for our body. Like without that we cannot survive. So like if I would ask you, ear is an like vital organ. So no, like people, like it's very important to have this organ too, but that's not a vital organ, because like without that even we can survive. Like vital organs like digestive tract or this digestive function, nervous system, these all are very, lungs, heart, these all are very essential. These are the vital organ. But ear and eyes, these are not that much essential, but they should be. So there's the difference between vital organ and organ. So now let's move forward. Present, they are named as like that. So first we take lungs. So if they would present lungs, so they would name as alveoli. Or if they are present in a liver, so that would name as Kupffer cells. Or if they are in microglia, which so there will be like neuron sites. These macrophagocytes will produce nitric acids. As we know, it's very toxic for our health or for our body. So what this will do, this will engulf pathogen which like protect, like which produce nitric acid against the bacteria, and then that will attach with the cell wall, like cell membrane, macrophagocytes. So like macrophagocytes will allow that to attach with the cell membrane of that. So then these macrophagocytes will make MHC. Now what's the full form of that? That is major histocomplexability, the full form. So this will be activate, like this will be their MSC, which will make order for the T cell. Like as we studied in the first line of defense, what was T cells, like here that this will send order to T cells to make antibodies. Bacteria is crossing F, like first line of defense, so it will enter the second line of defense. Then like when it will come to the second line of defense, so the MHC will order to T cells to make antibodies against that. So that's the work of MHC. So now But now, so here the response would be specific, specific immune. Like we noticed, like in second or in first both line of defense, there was non-specific, but in second defense it's non-specific, but still like that will be specific immune, because here MSC will send message to T cells, like it will order to that to make the antibodies against that. Interleukin, now what is that? Like there's the part, like there's the glycoprotein, and that is like hypothalamus. Now what is hypothalamus? Like it's a part of our brain which will regulate, like it will regulate temperature. So now here temperature would be regulated, so it will increase if like all of this process working, and that so there will be a rise in temperature, like it will be increased. So after that, like the pathogens which are present in a body will be killed, will be killed. Like how it will be killed? Because like if we are like rising the temperature, if the body will create a response, like as a result that will be rise in temperature. After that, like the pathogens will be killed, because like enzymes, as enzymes will will be denatured, pathogens will kill itself, pathogens will killed. So this all was the second line of defense. The dendrite cells, as we know, like these are also known as cytoplasmic projections. So like after maturation, this will move to the secondary lymphatic tissue, and it will be die because of the it will die, because as we studied, like T, like what was that, T cells, as T cells will stimulate or activated, as MSC will like order that to stimulate to kill that pathogen, so that will be activated, and after that that would be die. So the like dendrite cells will be die here because of the T cells, and it will stimulate, and the order will become from MSC, as I told the abbreviation before, MSC1. So now that dendrite cell is having short lifetime, like they are having very short lifetime, like maybe like they got 3 to 5 days, or like it's depend on them, like how long they want, like their work would be completed, the pathogens will be entered in the body, so that will be died, said like that's mean like they are having short lifetime in their body. And as in this, like as in the previous macrophagocytes, they were entering in that one. So the function of dendrite cells would be still same, like there will be engulfing of bacteria at the body of like that cell membrane, like it will enter at the cell membrane. And in that macrophagocytes, they were entering in that one, like vital organs, but in this they will enter like not in vital organs, they will go anywhere, like but except vital organs, as we discuss what are vital organ and organs, what's the difference between them. So they will not go in the they will go in the vital organ, sorry. So now let's study neutrophils. So what are these? Like these are non circular multi multi lobe or lobe multi. So this means these are 3 to 5 lobe, like the maturation approximately occur in the 3 to 5 lobes, and 50 to 70% of white blood cell would be present there. So that was all for today's video. Hope you like it.